There we go. All right. Thank you very much. We're going to get started again after the break. Hopefully everybody got a snack or some coffee or both. So it's my pleasure to kind of introduce this session, which is Building on Data Opportunities. My name is Flavio Roca. I'm from Oregon Health and Science. And my co-moderator here is Dr. Shishir Mathel from Emory. So we'll just get right into it. So we'll start with Genome-Wide Association Study. And we'll have Caitlin VanLith come up and talk to us. And then we'll have folks just stay on the podium, and we'll hopefully have some time for some questions after the talks.
Hi, everyone. Again, you've seen me throughout the conference, but my name is Caitlin VanLith. I'm a MD-PhD student at the Mayo Clinic in the lab of Dr. Lewis Roberts. On Wednesday, I gave a very similar talk, but that one was more patient-focused. This one's going to be more on the science side. Our methodology is going to be recruitment, opportunities for collaboration. Okay, we're going to skip the review of cholangiocarcinoma. As far as genetics go, there have been some really great presentations on this throughout the conference. Katie Kelly gave a really great overview that I'm not going to rival. But in our goals for this project in general, we want to better understand risk. We want to be able to identify biomarkers in germline DNA for cholangiocarcinoma development, and then be able to better identify targeted therapies. We've talked about there being some pretty good genetic links for cholangiocarcinoma, especially in the oncogenes. And so a lot of the variants that we see are common in these normal cancer-causing genes, both germline and somatic. But they're seen in a lot of different types of cancers, so we're not really sure at this point, due to low power in these studies, what specific variants or what specific comorbidities put these people more at risk for cholangiocarcinoma. So studies that have been done, these are the same slides as before. I'm trying to find different angles, but studies that have been done on specific oncogenes, more targeted explorations as opposed to general genome-wide association studies. And so our goal is to both validate these previous studies that were in small numbers of cholangiocarcinoma patients, and then also expand it to get higher power and really be able to see what those germline variants are, both in cholangiocarcinoma as a whole, and then the specific molecular profiles of intrahepatic, extrahepatic, PSC-related, and non-PSC-related cholangiocarcinoma. So this is a recent study that did look at germline variants and found some differences. And so these are genes that we're especially interested in our study, but we're going to expand beyond that in the genome-wide association part of it and have hopefully higher power. So as we go about this process, we've developed the International Consortium for the Genetics of Biliary Tract Cancers. In 2019, we underwent a discovery phase, and we're now working on the expansion phase. So that's the recruitment aspect of this talk. This is an effort led by Dr. Lewis Roberts, Dr. Catherine McGlynn, Dr. Chris Amos, and Dr. Manal Hassan. So we really have powerhouses on this project, and we were awarded a UA1 grant from the NCI, an intramural funding grant, in order to be able to recruit and then sequence all of these samples. In the discovery phase, we sequenced about 2,600 cholangiocarcinoma cases from 24 sites. All of these were in North America and Europe, and the bulk of our analyses were done on individuals of European ancestry. Yesterday, during the poster presentation talks, Young Hun Han from Baylor presented more specifically on that data. We're going to do a quick overview of what he presented. And if you're more interested in the specifics, he is the one that did all of the analyses for the discovery phase. As part of this study, we have two sequencing aims. The first one is to do the GWAS study on the global diversity array from Illumina. And so we're looking for really genome-wide SNPs. In the discovery phase with the 2,600, we did have some that came to light as being associated with cholangiocarcinoma. But we're hoping to increase the power of that because of the having to reach the genome-wide significance level. And as a second aim, we're doing whole exome sequencing through Regeneron in order to identify novel protein coding regions that may predispose individuals to cholangiocarcinoma. Let's see. Yeah, and then on top of these aims, we also have an epidemiological aim. So beyond these two sequencing aims, we're hoping to put together a robust epidemiological database of these patients. Valerie Gunchik, who's here, is from Vanderbilt right now. She helped develop this database with us. And we're also working hard to collaborate with Ciitizen in order to have a more centralized process for this information. This is, again, the 2,600 patients that we had in our discovery phase. And looking at overall cholangiocarcinoma cases in this cohort, we have one particular hotspot in HLA regions, HLA-DRB1 and HLA-DQA1. And Young-Han underwent more fine mapping to this to map it to a specific variant. As an overall summary of the study, he has an estimated heritability of 12%, and this differs slightly between intrahepatic, extrahepatic, PSC-related, and non-PSC-related cholangiocarcinoma. Again, as we stratify into these more specific groups, that 2,600 number is really coming down, showing the importance of the expansion phase of this project so that we can really replicate and have more robust results from this. He found 5 overall novel variants, or 5 novel variants, 1 overall, 2 in extrahepatic, 1 in PSC-related, and 1 in non-PSC-related cholangiocarcinoma. The 1 overall variant was that hotspot in the HLA-DRB1 and HLA-DQA1 region, and fine mapping was able to identify it to a specific variant with the other hotspots, the other points in the hotspot being related to linkage disequilibrium. And this is a novel variant. There's one publication that associates it with pleiotropic oncogenic effects, specifically cervical, kidney, prostate, and thyroid cancers. But more exploration is going to be needed into functional studies for this variant. It has a minor allele frequency of 13%, and we did find this to be a quite statistically significant variant, so we'll be on the lookout for it in this expansion study. Our goal is to sequence 7,000 cholangiocarcinoma cases over the next 5 years and 14,000 corresponding non-cancer control patients. We currently are working with 82 sites from 31 countries, including Cholangiocarcinoma Foundation. You saw us at the end of the hallway down here. At last year's conference, we were able to get blood draws from about 150 patients, and this year we're at about 100. So without being unethically coercive, I know that not everyone in this room has contributed, and I would encourage you to do so. This is going to allow us to look at even rarer variants. And we do have an emphasis on global genetic diversity and increasing representation in genetic studies. I'm sure that a lot of you already know about this, but certain genetic variants are more common in certain genetic regions, and past research has tended to be overwhelmingly white people of North American and European ancestry. So the specific global diversity array that we're using for the GWAS portion of the study has been validated in global regions, and the 1.8 million variants are really representing a lot of different populations globally. These are the sites that we're working with, and I also want to put in a plug for the consortium that we're putting together on the African continent. So as you can see, we're participating with or collaborating with a lot of sites in Africa, and Dr. Roberts and a lot of his collaborators and Matt Cooley have put together over the past couple of years the Africa Hepatopancreatobiliary Cancer Consortium. We're an R13 funded organization to put on a conference series of 3 years. We're hosting our 3rd one in August in Mombasa, Kenya. Stacy was able to join us last year in Accra. She presented on patient advocacy, and that was like a really important part of the conference. We had a lot of interest in that. And so we are looking for people who might want to join us, people who have collaborators in Africa, people who want to have collaborators in Africa. We have an amazing group of people that we're working with, including Michael Okello, who spoke yesterday. We have a great team of hepatopancreatobiliary experts from across the continent, and we're looking to increase capacity, increase collaboration, implementation science, and then we have some training aspects for it. So for the people who have not yet donated blood, we are in Grand Ballroom J until noon, so you have about an hour to get in there if you so choose. And if you don't have the opportunity to participate in this today, this QR code leads you to the Cholangiocarcinoma Foundation website, where you can submit information and get a blood draw kit shipped to you. Nellie Campbell is the main contact person for this aspect. She's been in the room in Ballroom J the whole week, doing a phenomenal job helping us out with this. So if you see her, please say thank you. And this all wouldn't be possible without an extraordinary team of individuals. And we really appreciate the Cholangiocarcinoma Foundation for financially and resource-wise supporting our efforts, both with the International Consortium for the Genetics of Biliary Tract Cancer and for the Africa Hepatopancreatobiliary Cancer Consortium. You've been a great partner in both of these things. So this is my email if you're interested in learning more about this study. If you have a biobank of cholangiocarcinoma samples and would be interested in participating, please reach out to me. If you're interested in establishing a biobank of cholangiocarcinoma patients, also please reach out to me. We have a lot of general protocols and more specific protocols as IRB templates for those. And then also if you're interested in the Africa Hepatopancreatobiliary Cancer Consortium, please also reach out to me. Thank you all.
So our next speaker needs no introduction. Dr. Juan Valle will be speaking about working with the new look of the ICRN, or the International Cholangiocarcinoma Research Network.
Thank you, Shashir and Flavio, for the invitation for this session. So I'm going to be talking about working with the New-Look ICRN. The ICRN is the International Cholangiocarcinoma Research Network, which is a global collaboration of research groups. By design, it is multidisciplinary and is working to improve knowledge about the etiology of cholangiocarcinoma, as well as prevention, early detection, treatment, and prognosis. You can see there our core mission is really to translate scientific discoveries into practice for cholangiocarcinoma patients. And again, you see this word collaborative research network, which is international. We do have, in fact, 340 members signed up for the ICRN. You may not see the detail in terms of the numbers, but you can see the map there in terms of the global distribution. I think you can see that it really is an international collaboration. So we've talked about the new look, so the obvious question then is what has changed? What we're doing is streamlining the ICRN a little. We've moved from 7 committees down to 4. So the first 4 remain the same. We have a data committee, a program organizing committee, the next generation committee, as well as the research committee. What we've done is to remove committees that we may be able to absorb within the foundation, for like membership and communication. The Master Protocol Committee, in fact, is now within the research committee. And a lot of the biopharma collaborations discussions that we're having largely are around the Master Protocol. So again, those have been incorporated. So it means that we're a little bit more streamlined with these 4 committees. And I've just highlighted here some of the programs that are underway within each of those. I'll focus on the data committee last, but the program organizing committee, of course, will focus on the annual conference, the consensus conference, which I told you about yesterday, as well as the Asia-Pacific conferences. And you heard that Stacy and others are increasingly engaged now in the Africa conferences. In terms of the next generation committee, I think we're all fantastically enthused to see people like Caitlin and others, that next generation of investigators and key opinion leaders coming through. So this is something that we really want to make sure that expands, grows, is supported with mentorship, webinars, young investigator meetings, as well as international initiatives. You know that we have a strong research program with our fellowship grants, the innovation grant, Cure Cholangiocarcinoma think tank, so C2T2, as well as master protocol, and then we have some additional research projects directly interfacing with industry partners. Just to quickly back to the data committee, you'll hear a bit more from the next two speakers about the Ciitizen and the Komodo platforms that we work with, so I won't talk about those any further, but just highlighting that from the data side of things, we're also working with GWAS team, CBio portal, and you'll see in one of my last slides the importance of harmonizing the data that we're collecting through our registries internationally. I'd like to just acknowledge our SMAB chairs, Scientific and Medical Advisory Board chairs, who kind of oversee the work of the ICRN, and thank them for their amazing work. In addition to rearranging the deck chairs, as it were, the philosophy has also changed a little. And I think here we've taken a little bit of a lead from how the ENSCA group works in Europe. So rather than having small committees in terms of size, we would like the committees to be open to people who actively want to become involved. And so the meetings may be much easier now that we can do these things on Zoom, but it'd be open to all members who want to be active within those, and then they'll report to the co-chairs, who will then bring reports through to the SMAB. Just in terms of efficiencies, at least one of the co-chairs, we would like to be a SMAB member. That means that they can then report to the quarterly SMAB meetings without having to have too much additional layering of reporting. And of course, from a foundation like ours, we feel that it's important to have advocacy represented in each of the committees. Equally, we want to make sure that this includes our nursing colleagues, so that it really is multidisciplinary and has the voice from everybody who is involved in the foundation. The Nursing Advisory Board used to report to the SMAB. I think they'll now directly report to CCF Executive, so that's a slight adjustment. It's not that we're moving them out, it's that in fact we're bringing them into each of the committees to make sure that their voice is heard through that process. So how to get involved? You've seen there's a big list of members. If you're not a member, you can join, and I think Ash is going to talk to this in one of his slides. While we're doing that, we're also going to refresh, just to remind people who may have signed up some time ago that we are changing. We want to have active membership, so we'll have a refresh and ask people which committee they want to be under. So have a think of which committees you want to be involved in. It doesn't have to be one, it can be many. The important thing here is about being actively involved in the community. And once people are ICRN members, then they're eligible to submit proposals to the data committee. And that's what I'm going to focus on for the last few slides. So the data committee is going to have kind of 3 tiers. There's going to be the ICRN members who are eligible to submit research proposals. Then there's the extended data committee, so this group of people will review the proposals and give some comments. They'll be coming back into the core committee, and the core data committee is going to be much smaller and a bit more streamlined. There's a little bit more of a demand on time because they will need to attend some decision meetings and decide on finalized proposals in terms of adopting projects or not. This process is still in draft, but you can see here that we will have an initial discussion with people who want to submit a proposal. This is not about saying yes or no. what we'd like to do, and we acknowledge that a lot of these people who want to put data submissions in will be young investigators, we want to work with young investigators to make it a good proposal so that the answer is yes. So sometimes there will be a bit of discussion around, have you thought about exactly what the question is and so on, so that we can really work with investigators. We want to be facilitatory. We don't want to be obstructive. And so the proposal submission will come in. We'll work with them to iterate the submission, the proposal, to make sure that it's ready for the data committee review. It'll then be submitted. It'll go out to the reviewers that I showed on the previous slide. And the role of the data committee is really to see where that particular project fits into the bigger picture. Obviously, we don't want to have two projects competing for the same topic. So if that is a risk of happening, then in fact we encourage people to collaborate and work together. But otherwise, the proposal will be accepted as a new research project. And then we go into the nuts and bolts of how we do this, what's required, what approvals need to be in place, and so on. Just not to scare you off too much, this is, again, in draft, but what the submission proposal is going to look like. We just want a little bit of information about you and your specialty. Then brief, think of this as an abstract. So we want a relatively short proposal, a little bit of background, what the proposal is, what's the question that you're asking, what's the methodology, and some references. So actually very, very straightforward. And then a little bit more in terms of where the process is up to. We are very keen that projects start and finish. So we very much have a view of where are you hoping to submit this, when are you hoping to submit this, what meeting are you aiming for, and where possible, get right through to publication. So nothing too scary, it's relatively straightforward. It's going to be a fairly seamless process, we hope. And this, on my last slide, just to highlight that what we're trying to also do is to align the data that we're collecting in the registries that you're about to hear in terms of Ciitizen in particular, align some of the data points with the ENSCA registry. That's the European Network for Study of Cholangiocarcinoma. They've got a well-established registry because the strength of data comes in the numbers. Being able to ask questions which are statistically powered is much more robust if we're all collecting the same information. So we are continuing discussions with the ENSCA group, and this will allow us then in time to be able to really have a number of publications coming out from the work that we're doing together. And with that, I'll thank you for your time.
Great. Well, thank you very much, Juan. We'll keep moving right along, and we'll have the Komodo update by Ash Patel, also from the foundation. Oh, we're switching it? Amanda? Yes. Perfect. Amanda Nottke, sorry. Come on up.
All right. Hi, everyone. Thank you so much to the organizers for this opportunity to speak with you all about the Ciitizen dataset, AKA the International Cholangiocarcinoma Patient Registry dataset. So I work directly with both Ciitizen and more recently with CCF, facilitating the utilization of real-world data. And today, here we go, I'll be first introducing the Ciitizen registry platform. Then I'll go into some more detail about how the data is organized and the current cohort that is available for research use. And then I'll spend some time going into some of the insights that are already being generated with this data. And then last but not least, speak a little bit about future directions. So the Ciitizen platform is a patient-centric platform. The way that this works is that patients find their way to the registry, almost always via CCF. They're invited to enroll. As part of that process, they give consent for Ciitizen to use the HIPAA right of access on their behalf to obtain all medical records relevant to their cancer care. So those records get collected and put in one place in the patient's account. They have access to it. They can share it with whomever they wish. And in the meantime, in the background, the Ciitizen machine learning-driven platform is sifting through all of these records and surfacing likely clinical variables, which are then confirmed via 2 rounds of human QA. And so at the end of this process, we have a normalized, standardized, structured output describing the cancer care to date, which is available to the patient as a summary, which again can be shared with whomever they wish, for example, for second opinions or clinical trial matching. But if the patient also consents for research use of the de-identified data, that data flows into the cohort that I'll be describing. Almost all patients consent for this use. We very much appreciate it. So now I'll talk a little bit about what the data looks like. So the data set is designed in order to answer important questions about cancer to support both research and clinical development. We think of the data of the variables in terms of 3 overarching categories. There's a set of things that describe demographics and clinical characteristics of the individuals and their cancer. There's things that tell us about what interventions were done, for example, surgeries, medication. And then we have variables that can tell us about outcomes, including efficacy, but also looking at tolerability, so for example, adverse events. And so each variable in the data set as part of this process gets normalized to one term, regardless of how it's described in the medical records. If it's referring to intrahepatic cholangiocarcinoma, it has that name. And so each variable has its name, it has the date or dates which it occurred or was observed, and there's also some linkage between certain variables. So using biomarkers as an example, we might have a BRAF positive biomarker finding. Well, that's linked to the Foundation 1 panel that was done. And that panel is connected to the site of disease from which it was sampled, whether it was the primary site or a secondary metastasis. Another example is adverse events being linked to the procedure or the medication that they're associated with. So taken together, this helps us ask, I think, a lot of really interesting questions about proportions, so how many individuals are experiencing what, as well as time to event calculations. For example, time from diagnosis to biomarker testing, or time from treatment start to progression. So taken together, these can help us understand things like the diagnostic journey, what's happening up and around the time of diagnosis, what's happening in terms of treatment patterns or treatment landscapes, what's being used, and importantly, what are the outcomes, the efficacy endpoints, and we can look at them through the lens of clinical characteristics as well as specific interventions. Last but not least, we can look at tolerability. And so this can support exploration of questions that are relevant both in the current state for helping everyone understand what potentially their best option is out of what is available, but can also power earlier stage questions about what is the unmet need and where can drug developers move in this space. So now I'll introduce the current cohort. So at the moment, we have 475 individuals who have consented for research use of their data, and that data is ready for research. So here I'm introducing some demographic and high-level demographic characteristics, and also doing a little call out about representativeness. So we want to keep an eye on the cohort as we're working with it, just characterizing how it looks relative to the general US population with cholangiocarcinoma. So you see the breakdown. The registry is a little bit more female than we might expect. The registry has a slightly younger age at diagnosis than we would typically expect to see. You can also see the geographic distribution. This is based on high-level zip code information, as well as whether individuals live in a metropolitan area or a smaller town or even rural. And you'll also note that we now have 1 individual from Canada in the registry, so we are officially international. All right, so looking into clinical characteristics, we took a look at anatomical subtypes, and of the individuals who do have an anatomical subtype in their data, not everyone does get a diagnosis more specific than just cholangiocarcinoma. But of those that do, we see the expected breakdown with the majority being intrahepatic, about 2/3 to 70%. Similarly, for those who are staged at the time of diagnosis, again, it's not everyone in the data set, but of those that are, we see what we would expect around the majority being advanced or metastatic. On the right-hand side here, we're also showing a breakdown of biomarkers split apart by anatomical subtype. So we can see, as expected, some of the biomarkers being more common in the intrahepatic subtype. Okay, so thinking about questions of treatment and what's happening in the real world setting that we can explore. One thing we want to keep in mind as we think about context is when people were diagnosed. So you can see that the mean median year of diagnosis is 2020, pretty recent, but as you look towards the lower rows in the table on the left, we have quite a few individuals who are long-term survivors diagnosed as far back as 2007. So I'll come back to this point a little bit on the next slide, but this does mean that not everybody in the data set necessarily has the same access to care at various points in their treatment journey, so that's something that we need to be aware of as we analyze the data. Now, in terms of what treatments people have received, we have a breakdown here of immunotherapy, specifically the PD and PD-L1 checkpoint inhibitors. You can see about 30% of individuals in the data set have received them, and the targeted breakdown as well. Last but not least, we do capture when individuals participate in clinical trials and see about 16% in the cohort. So the timing of registration, now this is kind of coming from the individual patient perspective. We wanted to take a look at when people are registering relative to their diagnosis. So the distribution that you see here, each horizontal green bar on this chart is a time for one person between their diagnosis and when they enrolled. So remember, we have people as far back as 2007. That's the bottom of this chart where we have this really long time. And as we look at the cohort overall, we see 50% are enrolling within 9 months of diagnosis. Coming back to my earlier point about representativeness, in order to make this data set the most useful for research and analysis, we want to have it as representative as possible. And the best way to do that is to have people enroll as soon as possible, as close to diagnosis. Now, all data is valuable. We're learning things from every single individual in this data set. But my call to action is, if you have not registered and you're interested, or if you're not sure if you've registered, come do it today. The sooner, the better. So now I want to talk briefly about what's already happening with this data in collaboration with various partners. So you may have seen these posters in the poster session. AstraZeneca, with collaborators, is looking at what treatments are people receiving in the first-line setting, how is it changing over time. Emory, Shashir's group, is looking at, for people who get surgical resection, what clinical characteristics are associated with time to recurrence. And we also have some ongoing work looking at access to quality care, specifically biomarker testing, and how that's being incorporated into treatment decision-making. So it's these two questions that I'll talk about over the next couple of slides. So coming back to this question of quality care, one of the metrics that we chose to look at access to quality care was rapid biomarker testing. So again, because we have points in time in our data, we can look what proportion of individuals are being biomarker tested within 30 days of their diagnosis. And we can split it apart by demographic characteristics, and we do not see a statistically significant difference along any of these dimensions. When we look at clinical characteristics, perhaps as expected, we do see that individuals diagnosed at early stage or that go on to have a resection end up having lower rates of biomarker testing within that 30-day window. Interestingly, we also see a lower rate for extrahepatic subtype, so we're continuing to explore this. And taking a little bit step back just to look at the overall utilization and impact of biomarker testing in this data set, we've done an exploration at the time of this analysis for 400 individuals. And we see over 90% have had tumor molecular testing at some point in their data set, which is a very impressive number. Of those who were tested, 37% have at least one targetable mutation identified. You can see the list of what we were looking for in that call-out. Of that 134, under 50% received a matched targeted therapy to their mutation at the time of data collection. So you might initially think, oh no, 50% of people have not received a targeted therapy. But given the depth and breadth of the data, we're able to look into the context of what's going on at the timing of a second-line treatment decision, which is when this would apply. And the first thing you'll notice is that the majority of these individuals, 60 of them, are not in the second line. So they're either early stage or they're still in first-line treatment. They're not yet eligible for a targeted therapy. Now, we see a handful of people who either received a targeted therapy for another biomarker, they had more than one, or at the time of their second-line treatment decision, the targeted therapy was not yet approved, so there was no way that they could get it. We do see a couple of individuals who, as far as we can tell, were eligible in the sense that they had the biomarker. The results were known at the time of treatment decision, but another type of therapy was selected. So we'll continue to keep an eye on that subset as we grow this data. So what did we do next? So keeping in mind that the platform is patient-centric and we can connect directly with registry participants, we wanted to reach out to the ones who had a targetable biomarker but had not received a targeted therapy at that time. And so after winnowing that down a little bit to those who have an FDA-approved targeted therapy, so at the time not looking at HER2, we sent an email out that described which biomarker was identified in the records, provided information that indicates you may be approved for an or eligible for an FDA-approved therapy. We can't make treatment decisions, of course, but please talk to your doctor. And finally, invited them to complete a survey. And so what we wanted to know was, did you in fact go on to get a targeted therapy? And we just haven't quite caught it up in the data. That was 3 of the 10 respondents. For those who hadn't, we wanted to understand if they had discussed it with their doctor. 2 of them had not at the time of responding to the survey. And then of those that did, wanting to understand a little bit better the rationale for the decision. And so again, this is something we will continue to do and also actively explore other ways to deliver personalized insights back to registry participants. So future directions, call to action. Again, if you're a patient or a caregiver, please join the registry. Our booth is outside. If you go out, it's at the far end of the tables, labeled International Patient Registry. You can also email support@ciitizen.com for assistance. If you're streaming in virtually, you can go to the Cholangiocarcinoma Foundation website and find your way easily to the registry as well. So next, researchers. We heard a little bit more about the data committee. Ash is going to speak to this more, so I won't say too much more, but first, join the ICRN. Next, propose a collaboration. And last but not least, for our industry potential partners, we would love for you to leverage the data set to help further advance research and development. Again, you can find our Ciitizen team outside at the booth or contact Nasha at the email you see there. Thank you so much.
All right, now we'll have Ash Patel giving the Komodo update, and then afterward, we should have time for some questions and discussion to really put all this together.
I want to thank Flavio and Shashir for organizing this session. I know it's almost lunchtime. I want to say thank you for all staying here. I'll try to make this a little bit brief and summarize what all of us are doing here with a lot of the data that we have access through the foundation. My name is Ash Patel. I'm the Director of Clinical Research and Data for the foundation, and my title of my talk is about navigating healthcare insights and leveraging the claims data that we get from the Komodo's healthcare map. Just quickly, I'm going to give a little bit of background of Komodo, how our relationship is with them, who they are, what they provide us, and then in terms of how do we utilize their data, how do we search on it, and the data sets that they give us. And also just kind of compare, making sure that all the researchers understand that, if it's representative of what we have in the U.S. Census data. And then I'll talk about some of the various projects that we have worked on. Also do a little plug of a poster we did for the conference as well, and some future directions. CCF and Komodo go back to 2018. That's when we had a formal relationship with Komodo. And this is one of the things where I think our fierce leader, Stacy, thought about how do we, get industry or other people that have this information, get them involved with the foundation. She saw this ahead of time, and this is when she brought Komodo into the play with the foundation in terms of getting access to their claims data. She wanted to figure out how do we use this information for the patients. This is a picture of her with Komodo's Chief Medical Officer, Ashwin. So Komodo's healthcare map, it's basically claims data, basically the patients that have insurance. Claims data is captured through various payers, hospital systems, and insurance systems. And it's very diverse. It's representative of the U.S. Census data set. It's de-identified. It's at patient level, but we do not know who those patients are. And then, again, their data sets is continuously growing. On a daily basis, they have about 50 million encounters coming into their platform, and access to basically information about 3.5 million healthcare providers in the U.S. population. So one of the first projects when we built this relationship was, what should we do with this? How can we help immediately to our patient population? So we decided to create the specialist maps for the patients. If you go out to our website, you will be able to find specialists in your state, and that's curated. I'm not going to go into the algorithm we use in terms of putting the list of physicians that are on that list. Melinda touched on it in her talk earlier this week. The other aspect of it is we wanted to figure out how do we connect with these patients. Because the data set is fairly fresh, we were getting the data from Komodo on a weekly basis in terms of who these patients were, where there were diagnoses, and then who the physicians were that diagnosed these patients. Melinda's advocacy team basically created the newly diagnosis program where they did outreach programs, try to identify those doctors, send them packets of information so that we could educate them in terms of some of these community doctors who have never probably seen or diagnosed CCA patients. So it was important for us to figure out where these patients are coming from and getting that information out, not only to the patients, but the physicians that are seeing these patients. And then from further on, again, we are also using this information to try to educate. We know there's a difference of the large academic centers versus the community settings. So we wanted to make sure that we had available information, resources that physicians in the community settings would have, so that they could also navigate the patients to our website and our advocacy team. So what is the Komodo data set? How do you search on it? And in terms of for the researchers, how do we ask questions against it? It's not really any information in terms of words and things like that. It's based on codes. We use the diagnosis ICD-9 and 10 codes. These are codes that physicians use for reimbursement purposes. When a patient comes into the hospital, they have to do billing, so the physician will code that this is the diagnosis I'm giving the patient, and then from there, if there's procedures that were done, they would provide a code for that so that they get reimbursed, and then if there's medications, they would have codes for those as well. And these codes for all of these different categories, these are standardized codes the U.S. healthcare system uses, and there are thousands and thousands of codes, and it's hard to figure out what to do with it. But with their system, it allows us to aggregate them, put it in context. For just cholangiocarcinoma, we could break it down into intrahepatic, extrahepatic, and it's all based on what was inputted by the physicians during the initial billing cycle. This is a plug for the poster session. This is a poster we did this year. What we're trying to do is we will try to do a publication on this, is to do a sort of a scope of what the data looks like right now and a snapshot on a high level with what we get from Komodo. We looked at the basic demographics as well as touched on some treatment patterns, and we looked at what were some of the diagnoses that the patients were seeing 1 year before their confirmed CCA diagnosis. And then we also looked at things like what was happening to them 6 months after. And the same thing with treatment. We looked at what kind of treatments these patients were getting 1 year ahead, and to see if there's a pattern there to identify what's happening to these patients. This slide is basically wanted to show you that the Komodo's healthcare map is really, this is the chart that you see here is their whole data set in terms of all cancer types, not just the CCA, but all cancer types, and how that matches against the US Census data. And as you can see, the blue is what Komodo's data set is representing, the green is the US Census, and it's fairly even and equal, not only geographically, but also with the race and ethnicity background. Without going into a lot of the details of all the results that for some of these studies that we've done in the past, these are just some of the questions that we looked at internally, as well as some of the ICRN members, to just kind of figure out, is this data set something that it could be valuable? Can we get some publications out and have an impact going forward? We looked at things like the proportion of CCA patients that have comorbidity of autoimmune disease, looked at patients that have a history of GI disorders, such as IBD. You know, one of the ones that a very interested conversation with a lot of people was a lot of CCA patients may get diagnosed initially as cancer of unknown primary origin, or CUP. In this particular, when we looked at the data, we found that about 23% of patients were diagnosed between 2018 and 22 that did have a CUP. So this is something that, you know, we want the investigators to understand, that this data is very valuable, and we want to be able to, for you to ask these types of questions. And then for these, if you have questions, we do have the data set available if you want to access it. So going forward, one of the things that we want to continue to do with our partnership with Komodo is that, you know, they're building new tools. Their data set is growing daily. And what we want to be able to do is find out what kind of questions our ICRN members want. We want to be able to target, and, so that they could not only be able to write tools for us, but getting access to the data sets that we want. So we're hoping that we get more involvement from the ICRN members. The areas that I think that we would love to get a little bit more input from is, you know, if there's data scientists that are part of your institution that may work with you, or biostatisticians, engineers, and analysis folks, we want them to be involved as well. They don't have to be pure clinicians. So if there's others in your department or in your organization, please have them get involved with our organization and ICRN. Future opportunities, one of the things that this is still in the pipeline. We've been talking about how do we build on what we're doing. You've heard from Amanda with the Ciitizen data set. You've heard from Caitlin about the GWAS studies. You know, what we want to do is start stacking this data. So yes, you could ask questions against these individual data sets that we have access to, but what we want to do is figure out possible ways to stack them, link the two data sets. You know, we're thinking about doing a pilot project with using Komodo and the registry data with the Ciitizen platform. The other thing that we've sort of modified this year is, I want to just say thank you to all the patients that have done the patient surveys in the past. That information is very valuable for us, but what we're trying to do now is actually revamp it. What we want to do with the patient self-reported survey data is to streamline it, have it research grade, and make it very friendly for the patient so that it's not thousands of pages. So what we're going to do is this coming year is to kind of work on that with our Canadian C3 group of trying to streamline that process and get that direct information from the patients. And again, we're also going to be trying to work with the GWAS team of trying to streamline their data and getting some of the clinical annotations from the medical records with the Ciitizen platform. So I encourage you again to sign up if you haven't done so already. My last slide here is really a call to action. I want to say, this past week, it's just been amazing. I over and over and over, I hear that this conference is not like any other conference. So I think, this is my way to say to you from the foundation is that for the industry, get involved, join the industry council. This is an avenue where, Juan talked about the ICRN and the mechanism of accessing the data. Well, through the industry council, you have access to that data. But in those settings, what we're going to do is it's going to be sort of a global question that it's not agnostic to just one company, but it's basically going to be for a common problem. Last year, we tried to address the DEI issue, looking at hotspots. So things like that where it's a common problem, those are the types of questions that we'll try to address as a group in the industry council. If you want to work with the individual companies, the Komodo or Ciitizen, please reach out to us. We'll make the introduction. We like to know what's happening so that if there's other projects that are going on, maybe we could leverage that relationship and make it streamlined for all the parties involved. For the academic researchers, if you haven't already joined the ICRN, I say please join the ICRN. Again, get others that are not even the CCA space, but maybe the bioinformatics folks that may want to work with us. Submit your own proposals. Juan talked about the process that we're trying to make it very smooth, streamlined process. The other thing I want to say is, for the young investigators, please get involved. The key thought leaders that are in this room and up here, they're very, very, very helpful, and they'll encourage you to succeed. So if you want to do projects, submit those projects, and you'll get some good input from the senior folks. And then again, patients and caregivers, thank you. Thank you for everything you guys do. All we could say is we're going to do our best, our part by, doing advancing the research as much as we can. The way that you could help us is join their registry, consent for things like the GWAS study, and then when we do the self-reported surveys, fill those out. They may not have an immediate impact on you and your specific issue right now, but I think one of the slides that I want to talk about is Caitlin's the other day. She said that they have had 2,000 patients that are enrolled in their first phase of the GWAS study. But when you start looking at some of the specific markers, there was only about 6 to 7 patients. So it's really important that you guys enroll in these studies because the numbers matter, and that's what we want you to do. It's going to increase the statistical power of making sure that we get really good publication results out there. Again, it's strengthen community and hope in research. So hopefully all of you guys work together with us as a community, and we really feel like there's a lot of hope in the research going forward. So thank you so much.
I realize that we're probably preventing you from lunch, but I think we have time for some questions, especially taking advantage of all these fantastic talks in this panel here. So maybe we can have the lights up and question time.
Angela.
Hello. Yeah. Thank you very much for all these presentations. I actually have a question for Ash. I'm very curious about your data, which is something quite unique for the US because you can track all these things because of the insurance companies. In Spain, we cannot really build this type of data or understand where patients are being seen. I was wondering whether you can find out how many patients are duplicated. What I mean by that is how many patients are a second opinion. How many of your patients seek a second opinion in the US? Can you, because if the data is anonymized, I assume you cannot really find that out. And I wonder whether you knew.
Yeah, I think Komodo does have because they're using a tokenizer, so they do have they know that it is a single patient. So they're probably a very small percentage. If we get technical about it, you could have some overlap. But I think for the most part, because they're using a tokenizer, when they're merging data from various different places for second opinion and everything, it does become a single patient.
Okay, because that would be something interesting to know, I think, and monitor. Thank you.
Mic too.
Sorry, is it you? Go ahead. So, Amanda, I missed some of your presentation, so you may have mentioned it, but I think the rate limiting step for all of these data registries is actually finding people to enter the data and the volumes of information. So are there models like CCF has like this army of volunteers who can probably assist? And the second is this, we just had a session earlier about the FDA and regulatory and quality of the data that can be used. Unfortunately, the FDA doesn't provide any guidelines about what this quality should be. So this can only happen if we interact with them and in advance prepare the data which they want, which I suspect that they don't know it themselves. Yeah. Thanks.
Yeah, thank you for that. So I'll start with your second question first. This is, of course, something we're very interested in and actively exploring. And so some things we're doing are working on validation of data quality by doing things like comparing to gold standard of manual chart extraction, and just continuing the conversations with sort of the appropriate, what are the audit trails and other requirements that the FDA would look for in this data. And to your first question, I think you were asking about scalability, so how we can increase the volume of this. And so, one thing that's really nice about the platform is that it is quite scalable. So we do have, the 2 rounds of human review and QA, but because it's supported by the foundation of this machine learning driven platform, the process is quite efficient.
I just want to add, before Eugene, I just want to add something more to that in terms of scalability. As you saw in the data that she presented, a lot of the numbers are skewed than you would expect from a national population because of the patients who are registering for it are coming to the foundation. And with 80–90% getting biomarker testing, that's just not the data if you look at the community as a whole. So again, charge to us at the foundation is really to get out there. Because there's maybe 25 institutions represented here or something like that. We have to get out to the other 300 institutions and small communities with our flyers, our cards and stuff, and get people from all over the country so that we can actually get a more representative population in the Ciitizen database. And one thing also I want to highlight is that database right now is a cross-section in time, but the plan is to prospectively continue to update it. So it will be a prospectively breathing database, which over time I think will become extraordinarily powerful.
Thanks for a great session. I had a few questions about the Komodo platform, but actually I think maybe a better way to put it would be to maybe inspire some of the young investigators in the room or online to help them understand the process through which they could submit a proposal. Because just to maybe plant a few seeds in their minds about what could be done here. I mean, you could use this, there have been multiple studies that have come out and shown that you can use EHR data for early detection or risk stratification of patients. Just looking at billing codes and saying, this patient is repeatedly coming in for cholangitis, maybe you should think about a diagnosis of a cholangiocarcinoma, for example. You can look at patterns of care for chemotherapy use, targeted therapy use, or radiation, or other liver-directed therapies. So what would be the process through which a young investigator who wants to try to answer these questions, what would they have to do other than submitting that proposal online?
Yeah, so I'll take that one. What we don't want young investigators to do is get too bogged down in which is the right platform. I'm confused. What data can I get from that? We'll worry about that. So the important thing is by joining the ICRN, then they are able to submit through there will be an online process with a very simple form that I showed. And then when we have that initial discussion with the young investigator, we can then work out which is the best platform. It may be data from both, it may be additional data that we need to get, but we will help to facilitate that process. So I think we're very open to just come with a question, and then we can work out the mechanics of which data sets we can access to answer the question.
We have a couple more minutes, I think, because we have I have a question for Caitlin. Currently, Laura's there. Oh, sorry. Go ahead, Laura. I didn't see Laura.
And tell me if this is too in the weeds, but I had a question about the adverse event tolerability that you kind of brought up, Amanda. Sorry, I should have told you. I'm looking at you in the database, and really there's been some touch points on it, even in our regulatory talk earlier about how difficult it is to kind of actually collect that data. In clinical trials, it's actually a whole lot of work above and beyond chart abstraction, but a lot of discussion with patients on clinical trials, and many patients who are here probably have that experience. And then to the point that Shishir just made about this is sort of a snapshot in time, and we want people early, but are we going to catch everything when the right time to look at that is? So my question is around how good how good is the data we're getting out if we're chart abstracting around specifically adverse events and tolerability? Probably not fantastic out of my regular notes if that were one of the cases. And then how many touch points with a patient directly may there be if they engage with Ciitizen, and are we doing anything with patient-reported outcomes to assess tolerability more deeply or maybe more accurately?
Great question, and it's never too deep in the weeds. Love getting into the data. So for adverse events, what we're collecting is clinician-documented association between a treatment or a procedure and an adverse event. So that's sort of the first bar. It has to be stated in the records. We don't make inferences about anything. The other thing that we capture is a variable that represents the impact of the adverse event. Did they have a dose reduction? Did they have a hold? Did they discontinue as a result of that? So that's sort of how the data looks in the data set. Now, I think to your point, it is limited by what's documented in the records. So we're not we're not picking up things that are not documented as adverse events. But because we track the dates of medications, I think there's the possibility for research looking into people that stop and we don't know why. They don't have a progression, for example. So I think there's opportunities to explore there as well. And I'm sorry, I'm totally blanking on the other part of your question. Oh, yes. Okay, so when somebody joins the registry, it's comprehensive retrospectively. So everything that has happened up until that point in time. But we do have the ability, and we do data refreshes. So at a certain point in time, we reach back out and collect all new records and then sort of fill in that delta in time. Yeah.
So I think as we go forward, understanding the exact data that's part of each of these databases is going to be key. So on that note, just two quick questions. One for Caitlin. Right now, is there any clinical annotation associated with these patients? And then also for Ash, for the Komodo data, is there any outcomes data, like treatment outcomes or survival data that we can get from the Komodo data? And then we'll ask one final question after that.
Yeah, that's a great question. Right now, the clinical annotation for the GWAS data is really being limited by our ability to abstract data. So working with Ciitizen and the ICRN is going to be incredibly beneficial to us. I am currently going through Mayo's patients, since that's something that I can do and I have access to, but getting data from other sites that are participating has been more difficult.
Particularly global. Ciitizen will help with the US part of it. The global part's going to be challenging.
Yeah.
Ash, in terms of that?
Yeah, for the outcomes for the Komodo, I think it's limited to what we have with the claims data. I know they're thinking about integrating with the Social Security death index data and all that kind of stuff. But again, that's why what we're trying to do is maybe do some stacking with the Ciitizen data, and that way we could get that information.
Great. Well, I think this has been a fantastic discussion. Yeah, go ahead.
Sorry, one final thing. I think when people think about, especially the young investigators wanting to work with these databases, they want to know how are they going to get the data, what is it going to look like? So can we just hear one line from the three of you? What does the export of the data look like in terms of analysis? Like how are we going to, like when you export a GWAS data, someone that says I want to do a project off the GWAS data or the Komodo data, I know a lot about the Ciitizen data, but we'll have Amanda say, but what does that look like? Very briefly.
Well, I can go first because I'm just going to echo what Juan said, which is it depends. So depending what resources and what capabilities you have in-house to work with the data, we can give you in sort of the raw data set, which is a tabular format, CSV. But there's also opportunity to have sort of support in terms of analytics, a little bit more help and hand-holding with working with the data.
With the Komodo, basically, they have a platform that's going to be their own sort of secured sort of box where you could play around with and do the data analysis and everything. But then if you want exports, they do give aggregate numbers and some tabular data, but not granular. So all the granular stuff stays within their secure platform. So they do have tools that are that allows you to do some analysis and everything within their own platform, and then get the sort of the summary data exported out into Excel, CSV, and all that.
We're still developing the sharing platform and pipeline, but I also imagine that we're going to be something similar to what Komodo has. I think individual level data, both genetic and clinical, is going to be probably something that would have to go through a further process, but summary level statistics for all of them exported in CSV files would probably be the first step.
Great. Well, thank you very much again. If folks have to get to lunch, but I'm sure folks will at the panel here will stay for a few minutes if you had some questions you want to approach individually or see them at the conference. Thank you very much.